Hello, this is Patrick O'Shaughnessy, and this is the 13th module in the series Statistical Methods for Analyzing Data Obtained in the Lab or Field. And here we'll be discussing the concept of a designed experiment and how this will ultimately allow us to do an analysis of variance. As with the previous modules, I'm showing here a module map. And now we're in a completely different set of modules. These are associated with the analysis of data for which you have multiple samples, meaning you have more than two samples that you want to compare. Again, a sample has, say, n observations within it. So of course, a one sample or a two sample test will be accomplished with a t-test, which was covered in modules 6 through 12. So if you know those, if you have reviewed them, or you are very familiar with the hypothesis test, how to conduct a t-test, then you can skip to this module here. Otherwise, you should review those modules prior to seeing this one in this progression of modules that ultimately get us to this analysis of variance approach. So now we're going to hopefully get to the point where we can analyze multiple situations at the same time to determine whether the mean of one is somewhat different than, or statistically different than the mean of some other. So these experimental situations that you are in, and there are of course almost an infinite different variety of them, but in the designed experiment approach, you, the researcher, have the capability to choose the magnitude of the x variable over some range of interest. So we've already seen this to some extent if you did the modules associated with regression analysis, where you could pick an x, say the RPMs of a motor, and you set it to a certain value, and then you see the pump rate associated with the pump that's attached to the motor, right? And then you dial in a new RPM and determine the pump rate again. So that's kind of the idea here. So this entire series that is associated with an ANOVA analysis is predicated upon the assumption that you, the researcher, have this kind of control over your experimental situation, and that the values of the independent variable, this x, are controlled by setting them in advance. You know what they are, and you're going to establish this as kind of the protocol of your experiment. And so when that is possible, then you can get into a variety of different types of experiments that are all collectively referred to as a designed experiment. And then this allows for a type of statistical analysis called the analysis of variance approach to determine differences actually in the means of these different levels that you have. Okay, let's kind of get started on this approach, and we actually already have, if you've reviewed the previous modules, especially those associated with the statistical tests and got to the point of two-sample t-test, you'll see that and recognize in this kind of concept that we've utilized this approach of being able to manipulate the levels, as the term is used in statistics, of the thing of interest. And these x variables in a t-test are often what are referred to as categorical, not numerical. So for example, let's say our x value is sex type. That is, again, not a numerical thing. And obviously we can split that between male and female. So there's our two groups. And let's say our outcome measure is asthma cases among 10-year-olds. So of course then we can control our choice of male and female subjects, and for example we choose 20 male and 20 female subjects, and we measure the number of asthma cases amongst those 10-year-olds and do a t-test to determine whether the mean level of asthma cases amongst males is different from, or statistically different from, those of the females. That's a t-test. Out in nature, X is fish type. Then the X was split between bass and trout. The outcome measure is the PCB concentration in the fatty tissue of the fish. So of course, in that case, we can go out and capture these fish, separate the bass from the trout, as long as we know the difference between the two of them, and then take them back to the lab, do whatever it takes to measure the PCB concentration, get, say, another 20 observations for bass and 20 for trout, and do a t-test. So we have control over this X variable, as you can see here, which is, again, in a categorical, whereas my previous example was RPMs, which is numerical, but again, we are setting it at an exact value in the case of controlling the motor from one RPM to another RPM. So categorical or numerical, it doesn't really matter as long as we split them into two different groups. Right, so in both cases, we're going to use a t-test to determine whether there is a significant difference between the means of these two levels of X. So then this begs the question of, well, what if I'm interested in 3 different fish types, bass, trout, and carp? What is the analysis approach that we can take if there are more than 2 levels of the X variable? And the answer to that question is that we have, again, the same kind of goal. We want to determine significant differences between the means of these levels of 3 or 4 or 5, whatever number of ways of splitting out X. And as we'll see, the analysis of variance approach will be the answer. Okay, so in terms of the terminology now, I've already used some of these words. We need to make sure we kind of hone in on it. First of all, of course, there's the experiment, which is the process of collecting the sample data one way or another. I'm going to talk about how to sample. Then there's the design of the experiment, and there are different ways depending on what it is you're trying to get out of them and or the constraints imposed upon your setup, for example. Then there's the response variable. We referred to this in the previous module on linear regression. It's the dependent variable, or the variable that's going to be measured in the experiment. And then there's the experimental unit. This is a new term, and it's simply when you get all set up, say in a lab, and you get a data value, what is that setup in a sense? And it's all of the different conditions that you have applied to get the data value. That's the experimental unit. It's that apparatus used to collect the measurements, typically. If it's out in the field, then it's the site in which you make the collection, and perhaps the environmental conditions, the meteorological conditions at the time, that sort of thing that define the experimental unit. Okay, more terminology. And again, I've already mentioned these to some degree, but we need to make sure we hone in on it, which is the factors. These are the independent variables. So you may say, well, we've already learned this in regression analysis. Why do we need to call it something new now? Why factors? And that'll hopefully become apparent as we move along here. The factors that may be related to the response variable, y. In the example of trout versus bass, the factor actually is fish, or fish type. We had 2 levels of them, and that's the next word. Levels are the intensity settings, is the best way of putting it, because again, intensity could be numerical or it could be categorical. In the case of the fish, it's 2 fish types. They are at 2 levels for the factor fish type. For the previous experiment, the factor is sex at 2 levels, male or female. So you get the idea? So a t-test consists of 1 factor at 2 levels. They are, you can't compare them unless they have something in common. 2 different cornfields, well, they're both cornfields then. 2 different fish, well, they're both fish types then, right? So you have this 1 factor that is the commonality split out into various levels. Again, a t-test, we didn't use these terms, but it's perfectly appropriate to call a t-test a 1 factor two-level experiment. Okay, now treatments. You'll see this term pop up, especially when we talk about ANOVA analysis. They use it all the time. Treatments are the particular combination of the levels of the factors, you see factors plural, involved in the experiment. So the experimental conditions set up for a trial. Now I mentioned that in terms of the experimental unit, but there's going to be different conditions because you're going to change these levels. Maybe a high and low temperature, along with maybe a high and low relative humidity, for example. In that case that I just described, there were two temperatures and two relative humidities. 2 times 2 is 4. There's 4 different conditions that I need to set up in my lab if I want to get at all possible combinations of the 2 temperatures and 2 relative humidities. Those 4 combinations are what are referred to as the treatments. Now, the number of treatments for a one-factor experiment is simply the number of levels. There's no difference. In that case, all I care about is temperature, not relative humidity, for example. And I'm going to split temperature out into 2, a high and a low. There's no other way of splitting it out. So the number of treatments equals the number of levels for a one-factor experiment. Okay, let's get an example here to kind of hone in on these different terminology types. So it's an experiment conducted to determine the effects of cutting speed and mortar type on the concrete aerosol concentration produced. Both cutting speed and mortar type can be controlled to specific levels. Good. So an experiment was conducted during which the cutting speed was set at 2,000, 3,000, or 4,000 RPMs, while 2 different types of mortar were selected, hard and soft. The experiment was set up in an enclosure and repeated 3 times for each condition, during which the aerosol concentration was measured with a gravimetric filter sampler. Okay, so this is an experiment in terms of exposures, say, to workers while they're cutting mortar with a certain type of cutter. So we can set this up in a table and encourage you always to do this whenever you're designing your own experiment. How can you tabulate it like this? So we see from the description that there were 3 different cutting speeds. So we need to have 3 columns associated with cutting speed. And there were 2 different mortar types, hard and soft. So we have these 2 rows. This now helps us to set up exactly what we're going to do in each one of our experiments in terms of the experimental unit being any one of these repetitions of this particular setup that we have for getting the data we need to get. And notice there it says they're repeated 3 times for each condition. So you can see that here. Now these numbers vary because ostensibly you should randomize which of these conditions you do first and then which one you do second, and that's what's going on here. 1, 2, 3, 4, 5. That's what's happening. You should not just do them sequentially through. That's often impossible to avoid in many experimental situations, but you have to be honest about that because it can induce a bias. Things can change as time goes on. Maybe the cutter gets duller and duller over time, and therefore more and more dust is being generated just because you finally get to the last experiment here and you have a dull bit that's starting to really just crank out a lot of dust for whatever reason. Maybe it's just the opposite. Maybe you have more up front, but that's just an example of the bias that can happen in an experiment that's run sequentially like this. Okay, but take a good hard look at this. We have 3 by 2. How many different combinations? 6. 2 times 3. It's 6 different combinations. I have to set it at 2,000 and versus a hard mortar. Set it again at 3,000 versus hard, 3,000 versus soft, etc. These are the treatment combinations. They're the combinations of the levels of each type. Okay, so what are the factors? Well, in this case, as you can imagine, again, now you have a picture in your mind of it. They are the two main things that are being adjusted: mortar type and cutting speed. So how many do we have? 2. What's the response variable? We're going to measure something. In this case, it's going to be the concentration resulting from the cutting of the mortar. What are the number of levels for each factor? Well, for mortar type, hearts off, 2, cutting speed, 3. And what about the experimental unit? Well, again, that's one repetition of the experiment using this chamber that we're going to do this experiment in. The number of treatments, as we just discussed, well, you just multiply your levels. 2 levels times 3 levels equals 6 treatment combinations. And again, you can see why because of that graphic we just looked at. So what's the total sample size? You just multiply the treatment combinations times the number of repetitions per treatment. So you want to go all the way back to the beginning. 2 levels times 3 levels times 3. Okay, so we have 18 total samples. This is what you should go through in the process of designing your experiments. You know how many samples you're going to need to take, and then you may need to know, how much it's going to cost per sample, how much time it's going to take, etc. This is a 2-factor experiment. It's no longer a 1-factor experiment. It's not even 1 factor at 3 different levels. So this is getting way beyond a t-test. I can't apply a t-test to this no matter how hard I try. You can say, oh, mortar type, there's 2 of those. And I can compare just between hard and soft, regardless of cutting speed. But we'll find through the ANOVA analysis technique that there's a better way to do it than that. Okay, steps in designing the experiment. Of course, you're going to first select the factors to be included in the experiment. But you're also going to identify, what parameter are you interested in? What's the object of the study? Typically, as it says here, it's the means. Remember, a parameter is the attribute of the population. It can be typically either the mean, mu, or the standard deviation, sigma. And there are tests for sigma. But when we talk about hypothesis testing, the hypotheses were associated with the mean. The mean equals 0, the mean is not equal to 0. So that is the typical scenario. Step 2, you're going to choose the levels for each factor. Again, you need at least 2. And depending on how many levels you have for each factor, that will determine the number of treatment combinations you have, as we just discussed. And finally, recall that a 2-level factor comparison, again, involves the use of a t-test. I should say a 2-level single factor comparison involves the use of a t-test. Anything beyond that, and we're going to have to use more sophisticated techniques. Step 3, determine the number of observations to be made for each treatment. And they should be the same. Hopefully, it's what's called a balanced design. When that happens, then you can then determine the total sample size. And finally, plan how the treatments will be assigned to the experimental units. And that's what we're going to discuss next. Okay, so what is the plan you're going to use to collect a sample for one of the experimental units? And you're going to repeat it, hopefully exactly the same way for all subsequent ones. This is called the design of the experiment. And, right off the bat, you could think of, well, am I going to collect randomly or am I going to collect sequentially, or what they call kind of a quasi-randomly? And what is the sample size needed? Well, it's best to increase the sample size as much as possible while also reducing the variability between samples as best you can. And again, the variability is often a factor. It's inherent to the system that you're measuring. So you don't have much control over it. The only thing you can control in terms of variability is the accuracy or the precision, to be more specific, the precision of your instrument. So recall that the standard error determines the ability to observe statistical differences between the means of the factor levels in the t-test. And the standard error consists of sigma over the square root of n. So you can see just off that simple equation that if you increase n, you're going to increase the number in your denominator, and therefore you're going to decrease the standard error, and that increases the power, your ability to distinguish one population mean from another population mean. Now this whole idea of power again and how you compute it was discussed already and takes more sophisticated software than Excel has to calculate. Okay, now we're going to talk about some sampling techniques. Most common, or at least advisable, way of sampling is to sample randomly. And it's called simple random sampling. It's used to compare so many treatments, you got P of them, in which every element of the population has an equal chance of being selected. That is the goal of it. You're trying to ensure that in your sample you have some of the low values, you have some of the high values, you have the ones in the middle that are bunched in the middle. So you can kind of reproduce the distribution, if you had to, of the population that you're sampling from. So it's a design used when sample observations can be randomly chosen from a population, essentially an infinite number of times, and you don't have any other factors that influence the selection process. So for example, comparing 3 fertilizer types applied to 3 cornfields. You then are going to randomly select the heights of, say, 50 corn plants in each field. Again, how you do that in terms of the randomization process, there's probably some tricks to that, but you can imagine some. We discussed earlier how you could just go out with a blindfold on and just pick the first 50 that you touch. So again, the goal is to sample in a way that results in obtaining measurements from the entire population that spans its distribution. You don't want to have, as a result of all this, to have most of your samples on the high end. You want to compare the heights of students, University A versus University B. You don't want to end up with a sample that contains most of the basketball team. Okay, that would skew your reality of the heights towards the high end. So we want to make sure that we avoid that sort of a bias in our sampling. To randomize it as much as possible.